When researchers created Dolly the sheep by cloning, it was the first time they had had such success with a technology known as somatic cell cloning or somatic cell nuclear transfer. That was in 1996, and in the nearly 3 decades since then, researchers have successfully tried and tested the same technology on a further 20 mammalian species. Cloned mammalian animals often suffer from damage to their muscles and/or other organs, and that dramatically reduces their life expectancy. Cows have so far had the best chances of staying alive. Primates have had the lowest likelihood of birth and survival. In early 2018, however, it was reported that two cloned macaque monkeys, Zhongzhong and Huahua, were born the previous November at the Institute of Neuroscience of the Chinese Academy of Sciences in Shanghai. The international research community considered it a sensation. Now the same institute has published a new success story. Researchers at the institute cloned a rhesus monkey. The male is said to be healthy and happy and already 2 years old. Their research has been published in the science journal Nature Communications. Is a cloned rhesus monkey a breakthrough for science? The study and the technology they present tell a very special and biologically interesting story, said Rudiger Bayer, a stem cell and embryo researcher at the German Primate Center. But I doubt we're seeing anything fundamentally new or anything that will find a broad application in the science community. Bayer said the researchers in China use a technique that only a few laboratories in the world could replicate. They created a combination embryo, one part cloned and another part not cloned, said Bayer. He added it was hard enough to develop a fully cloned embryo, implying that this technique was even more complicated. How does cloning work and why is it so difficult? The biggest challenge in cloning, that is, creating an exact genetic copy of a complex life form, is the clone's survival. Most cloned animals have poor health and short lives. As with human animals, all animals and other living organisms are made of somatic cells. A somatic cell is any cell that is not a reproductive cell, such as sperm and egg cells, which are called germ cells. In somatic cell cloning, researchers plant the cell nucleus of a somatic cell, such as a nerve cell, into an egg cell that has had its own nucleus removed. When you do that, the egg cell can reset the cell nucleus of the somatic cell so that the entire construct behaves as though the egg were fertilized, explained Bayer, and from that you get an embryo. The fertilized egg cell begins to divide, creating exact copies of itself. But the more the cells divide, the more they differentiate themselves from each other, and their functions they become skin, heart, muscle, and nerve cells. The genetic information in each cell nuclei is the same, but not every gene gets read. Think of genes as the words or pictures in a Lego manual. If you skip a page, you end up building something different from what's pictured on the box. That is how different cell types are formed. But with this technique, the egg cell reprograms the implanted somatic cell nucleus to allow all the genes to be read the same. As when you reset your phone to the default or factory settings, the cells start afresh. That is the theory anyway. It doesn't always work very well, said Bayer, alluding to the low efficiency rate in cloning. Is it especially difficult to clone primates? When it comes to efficiency rates, the current study is no exception. Of 484 implanted embryos, only one was born alive. But it's almost impossible to know, said Bayer, whether that one successful birth was due to the new technique or pure chance. In principle, said Bayer, it is not fundamentally more difficult to clone primates than mammals. There are only very few institutes in the world that can even conduct such experiments with primates. So, in my opinion, the fact that cloning remains difficult has more to do with the fact that we've so little experience with cloning monkeys, he said. Keeping monkeys for research is more intensive and expensive than keeping mice, said Bayer, so slower progress is made with monkeys than with mice. Why clone monkeys at all? There is considerable interest in cloning monkeys because non-human primates, such as monkeys, are biologically similar to humans. Researchers hope to use monkeys more in medical research to treat Alzheimer's and Parkinson's diseases. If researchers could conduct experiments on groups of genetically cloned monkeys and compare the results to the same tests on non-cloned monkeys, they believe positive medical findings would transfer better to humans than if the experiments were conducted only on mice, for example. As a PhD candidate, I couldn't imagine that one would want to clone monkeys, said Bayer. That was a red line for me. Monkeys are too similar to humans, too sensitive to pain. But Bayer said he sees things differently now and that there are situations in which cloning monkeys may be appropriate, such as for untreatable diseases that cause great suffering. One goal is to develop gene therapies for visual impairment, deafness, heart disease, and metabolic conditions. But no one knows, conceded Bayer, whether cloning monkeys will help us find the desired treatments. Subscribe to watch more videos like this. Turn on notifications and leave a like to help the channel out. Thank you for watching.